Hello everybody, welcome to part 2 of the digestive system. We're going to continue through the digestive tract components. If you haven't already, make sure leave a like on the video, drop a comment, subscribe. Any of those things really helps me out. Okay, so we talked about esophagus, we talked about the 4 layers, we talked about kind of the general structure of everything. And now we're going to be continuing that kind of same tract, talking about the stomach. So functions of the stomach. So we continue the digestion of carbohydrates that started by amylase in the saliva of the mouth. Remember, amylase digests carbohydrates. If you have not watched the digestive enzymes video, that will cover that as well in the oral cavity. The stomach's job is to convert that food bolus to something called acidic chyme. That's the key word that you'll look at. Chyme is produced in the stomach. It also starts to digest triglycerides or fats by lipase, and it also starts the digestion of proteins with an enzyme called pepsin. We'll talk a little bit more about pepsin in a little bit. This is just a little way to remember pepsin. We'll cover this a little bit better in the future. Pepsin is released by these cells called chief cells. They secrete something called pepsinogen, and then the stomach acid turns pepsinogen into pepsin. We remember that to say the chiefs drink Pepsi. So we'll talk about that again in a minute. Here is the overall anatomy of our stomach. We have a couple different places. So we have right where the esophagus goes into the stomach. That is called the cardia. The top portion is known as the fundus. We have the body, which is the main chunk of it. And then we have the pyloric or pylorus portion of it. And then another sphincter called the pyloric sphincter, which continues the food. It will take the chyme and put it into the duodenum or duodenum, however you want to pronounce it, which is part of that small intestine. Okay, we just want to remember fundus up top, cardia where it enters, body is the main part, pylorus is where it leaves. We also have these folds, these things called rugae on the edges. This is so that it allows our stomach to expand. So kind of like how, like if you have a balloon and you deflate the balloon like a big yoga ball or something and deflate it all the way, it's kind of like wrinkly because, it's fully deflated. But then when you fill it up, all those wrinkles go away. Same thing with the stomach. That's what these rugae are designed for, to allow the stomach to expand. Here are our 4 layers. We have mucosa, we have submucosa, we have our muscularis externa. We have 3 layers here. Typically, we have a circular and a longitudinal layer. We have an extra layer in the stomach known as the inner oblique layer of muscles, and then also our adventitia and serosa. Here's just another picture showing all the different parts of our stomach, the rugae, all of that broken down, as well as here the mucosa, submucosa, muscularis, and serosa, all showing right there, just giving us a little bit of an idea what these looks like. We're going to jump in and talk about the mucosa. Here's all the stuff that we're going to talk about. Gastric pits, surface epithelium cells, mucous neck cells, parietal cells, chief cells, enteroendocrine cells, as well as gastric glands. Okay, we have the gastric pits. So gastric pits are small openings in the stomach lining that actually lead to the gastric glands. This is where, essentially, it's going to be these little pits, these information pits, where the food, everything that we get, goes down into there, and it activates a sensor saying like, hey, there's food in the stomach, there's stuff going on, there's stuff that we need to digest. And that's what kickstarts the release of the enzymes, the chemicals, to help us digest our food. We have the surface mucous cells. So these surface epithelium, those outer layer of cells that line the stomach. These surface epithelium, also known as mucous cells, doing what epithelium does. Its job is to protect the stomach lining and they are going to secrete a highly concentrated alkaline mucus. Think about stomach acid is very acidic, a very low pH. So they secrete a mucus that is very alkaline or basic in order to create a barrier between the actual stomach itself and the stomach acid. Make sure that it keeps itself safe. And it's made up of simple columnar epithelial. Most of the time when we're dealing with mucus secretion, we typically are looking at more of a columnar type shape, but not always. Like the esophagus secreted mucus and they had mucus cells, and that was a simple squamous epithelium. So, but simple columnar epithelium here in the stomach. The mucus neck cells, so we're continuing down inside of these gastric glands. We get these mucus neck cells. Just like their name, they continue to secrete mucus to coat and protect the stomach. So it's more alkaline mucus being produced in these cells. Now we get into some of the goodies, the parietal cells. Parietal cells, which are found deeper in these gastric glands, right here, parietal cells, along with chief cells, parietal cells and chief cells. In the lab portion, you're going to see a lot of pictures with parietal cells and chief cells in the same pictures, okay? So I think I have a couple, yeah. So you have like parietal cells and chief cells look very similar to each other. These parietal cells just have a little bit of a darker pink stain on them. So parietal cell, parietal cell, parietal cell, and these are chief cells. Parietal cell, parietal cell, parietal. The rest of these are chief cells, okay? Anyways, the parietal cells secrete two things. The first thing, they secrete hydrochloric acid. They secrete our stomach acid. It makes the pH about 1.5 to 3.5. Around 2 is what you typically say for the stomach. It's around a pH of 2. And at this pH, a couple of things happen. This is the pH where proteins will denature. If you think back a little bit, we talked about how proteins are made in biochemistry, how they all come together with peptide bonds and they fold all together and all that good stuff. At extreme pH differences, that actually causes them to unfold and break apart and break down. Denaturing of a protein causes it to unfold, causes its bonds to let go, and breaks down the proteins. We get pepsin activation. We also have breakdown of plant cell walls. So if we eat our vegetables, and remember plants are a little bit different, where instead of just like in eukaryotic cells like we are, we have a phospholipid bilayer. Plant cells are a little bit more square, and they actually have a secondary coating called a cell wall. So our stomach acid can break that all down for us, as well as it kills a lot of the bacteria that we don't want in our bodies that may or may not be on our food. So that's hydrochloric acid. We also release something called intrinsic factor. The biggest thing that you want to associate intrinsic factor with is our vitamin B12. We need intrinsic factor in order to absorb vitamin B12. Vitamin B12 is essential for making sure that our DNA is kept healthy, as well as producing red blood cells and that kind of stuff. And we'll talk a little bit about those conditions with B12 and when we get towards the end of this portion, we get to the clinical application portion. Hand in hand with the parietal cells, we have the chief cells. Remember what we said? The chiefs drink Pepsi. I'm going to say that a couple times because I want you to remember that because it helps with a lot of test questions. So the chief cells secrete an enzyme called pepsinogen. This is the inactive form, meaning that it does not do anything right now. Once it hits that super low pH of the hydrochloric acid, it gets activated and it turns into pepsin. Pepsin helps to digest proteins. Okay, pepsin also has another job that it actually, while it is activated by the super low pH hydrochloric acid, if a pepsin runs into another pepsinogen, pepsin can also turn that pepsinogen into more pepsin. So it does what's called a positive feedback mechanism, or a positive feedback loop. We don't see very often positive feedback loops. The only other one that we ever really talk about is oxytocin in regards to giving birth, typically, which you'll talk a lot about in physiology in the future. Mostly we talk about negative feedback loops, which we will have seen a lot in biochemistry, and we will also see a lot of those in physiology. But pepsinogen is one of our very rare positive feedback mechanisms. Enough that if I was a professor, I would probably throw that in as a question, just because I think it's a really good one to ask. Okay, chief cells also release some lipases, which start to digest the fats. Finally, we have enteroendocrine cells. Break this word down. Entero, meaning inside. Endocrine, we talk about the endocrine system. We're talking about hormones that go off and act on other stuff. So these cells produce and release hormones into the lamina propria. If you remember, the lamina propria is part of what? The mucosa layer, right? It's one of those three parts of that first layer, the lamina propria. Some of them that it releases, we have serotonin, histamine, and somatostatin. These ones can act as paracrine hormones. Paracrine, para meaning next door, crine meaning referring to endocrine. So para affects nearby or neighboring cells. Another two hormones, somatostatin as well, and gastrin can also act as endocrine hormones. So those travel through the blood to farther away targets in order to stimulate other things. And we'll talk. You'll talk a lot more about gastrin, somatostatin, all of these in physiology courses. But I just want you to know that gastrin specifically is what goes and stimulates those parietal cells to release hydrochloric acid. And this is more future knowledge. You don't get this specific in this course, but you will in the future. They are released from G cells from the gastric antrum and the duodenum. Here are those gastric glands. Essentially, the gastric gland is the whole chunk down here containing all of this stuff. That's why I kind of left it to last. The gastric gland is what contains all of this other stuff. It has the chief cells, the parietal cells, the enteroendocrine cells that produce all of those things. Clinical application, I mentioned that intrinsic factor. This is what we want to talk about. So if we don't have intrinsic factor, we cannot absorb vitamin B12, and we have issues with the making of erythroblasts, also known, red blood cell makers, and we get something called pernicious anemia. What pernicious anemia is essentially that our red blood cells are not being built correctly, so more of them are being destroyed, so we don't have as many red blood cells. That's what anemia is. Anemia is not enough healthy red blood cells. So our body ends up destroying a bunch of those red blood cells because they're not healthy, and we have consistently low red blood cells, which, as we know, red blood cells are necessary for us to survive and keep going, so that becomes an issue. The two big causes for not enough intrinsic factor is it can be an autoimmune condition, so like our own immune system is attacking the parietal cells, or it can also be age-related. Autoimmune condition or age related. Older people have less acidic stomachs. I should say older people have less effective parietal cells, so less intrinsic factor. So age, it's either an autoimmune condition or it can be age related. Continuing on to the small intestine. Let's look at the functions. This is the final site of digestion. The absorption of the nutrients happens here, which again we talked about. This is the whole point of our GI tract. We need nutrients because we need to convert those into energy to survive. And then we also get a bunch of endocrine secretion. The kind of macro, the gross anatomy. That's what I should be putting. Instead of macro, I should put gross anatomy. I prefer macro. It makes it sound nicer. We have 3 segments. We start at the very top, the duodenum or duodenum, however you want to pronounce it. It's not wrong. You'll hear it pronounced 30 different ways. It's fine. Don't worry about it. This is the very first portion of the small intestine. The pyloric sphincter, remember this end region of the stomach is called the pylorus. And this sphincter is what connects the stomach to the duodenum. Now do you remember what we call the food with all the mixed up acid and everything inside of the stomach? That is that acidic, starts with a C, chyme. The acidic chyme in the stomach. So that acidic chyme is going to be pushed out into the duodenum. It will eventually continue through the jejunum and then eventually into the ileum. The ileum then into the large intestine. And we'll talk about that when we get there. So, layers of the small intestine. We're going to break these up a little bit as well because each part of the small intestine has a slightly different responsibility. Overall, the idea is absorption, but they're broken up into these 3 parts because they do slightly different jobs. So we have the mucosa. There are tons of secretion cells in the mucosa in the small intestine because this is where we're going to finish digestion. This is where we're going to break a bunch of more stuff down and absorb a bunch of stuff. Of course, we have our submucosa. We have the muscularis externa, the circular and longitudinal layers, and then classically the adventitia and serosa, which we'll pay more attention to that at the very, very end. There's a little chart for you guys. This is a little bit of extra information. We're going to cover all of this in future slides. This is something that the professor had that I borrowed, that I took the baseline of this information from. They had this here, and I'm not really going to focus on this. I broke it all down in the future slides. So, okay, so the small intestine mucosa layer. There are 4 things that we're going to talk about here within the mucosa layer. First things first are these structures called plicae circularis, or these circular folds. I thought I had a better picture of these guys. Anyway, similar to when we talk about surface area, and we know we want to do absorption. In order to absorb things effectively, we need a lot more surface area. So these structures are going to allow us to increase our surface area in order to get the maximum out of absorption that we need. I thought that I had... Okay, I covered it a little bit later on. Okay, it's between the 2. Okay. Well, we're going to come back to these guys, the plica circularis, the villi, and the microvilli. We're going to come back to those, and I'll have a couple of good pictures and stuff for those. We also have these intestinal glands, also called crypts, or crypts of Lieberkühn. Between the villi and the epithelium, between the villi, and they're going to contain some stuff in there that we're going to talk about again in a little bit. Some of the cells of the mucosa. I have specific slides for all of these. So enterocytes, goblet cells, Paneth cells, and enteroendocrine cells. And then we've mentioned these before when we talked about the immune system. We also have Peyer's patches in the lamina propria. Here are our Peyer's patches. Those are connected to our immune system in the small intestine. And let's talk about these specific cell types. So enterocytes. Enterocytes, these are our intestinal absorptive cells. They have columnar epithelial cells with microvilli. Remember we talked about villi, microvilli, how it increases our surface area to allow for absorption. We talked about that way back, talking about epithelial and also like the actin structures. So villi, these are responsible for a lot of our absorption. Goblet cells secrete mucus, protection and lubrication. That's about all I'm going to say about those guys. Paneth cells, these are base cells of those crypts, or those, what's the other word for the crypts? The intestinal glands, the glands or the crypts. These guys' these are part of our defense system. They secrete lysozyme as well as defensins. The professor that I took this from only mentioned lysozymes, so make sure that you know that one. But they also secrete another chemical called defensins. These have antimicrobial peptides, so essentially they locate and they attack and digest and destroy bacteria within the gut. Okay, now the big ones are enteroendocrine cells. This is where we have a bunch of different hormones that are secreted to help with our digestion and help us know what we're digesting and when to release them in order to digest things at the right time. Because we don't always have food in our stomach, right? The two that we'll focus on are cholecystokinin and secretin. In future courses, I put information in here for them, but they didn't mention them. This professor didn't mention gastric inhibitory peptide or motilin. In the future, you will talk about those when you get a little bit deeper into the GI tract, okay? So cholecystokinin is secreted in the duodenum and the jejunum from these cells known as eye cells. They are released when there are fats and partially digested proteins in the small intestine. So when we have that acidic chyme go through the pyloric sphincter and into the duodenum, it says, hey, we got fats, we have proteins. It releases cholecystokinin in order to go and stimulate the gallbladder to release bile. Remember, the gallbladder emulsifies fats. It doesn't digest fats, it emulsifies them, which means it mixes with the fats and kind of pulls the fatty acids away from each other so that they can be digested easier, because fat kind of likes to cling and stick together. We also get secretion of the pancreatic enzymes, the lipase, the amylase, the proteases, all of those guys that are going to break down the stuff that is in our food. We also then have secretin is the other one that we really want to focus on. These are released by S cells in the duodenum, and its job is to neutralize the stomach acid in the small intestine. Because you think the stomach is super prepped and prepared to handle that acidity. The small intestine, not so much. So as soon as we have the acidic chyme go into the duodenum, it signals that, hey, there's acidity here, we need to take care of this. Secretin is released. It goes and it tells the pancreas, hey, this is too acidic, we need to make sure that we neutralize this so that way we can handle it and deal with it and not get injured. So it releases a bunch of bicarbonate fluid, which neutralizes that stomach acid to allow it to go through the small intestine. gastric inhibitory peptide, as well as motilin. You can pause to read each of those if you want. This won't be tested in this class specifically. In future courses, though, you will see it a little bit more. We're going to cover this mucosa, submucosa layer, and then we'll jump back to all of the plicae and the villi stuff. So within this, the submucosal layer, we just have these glands called Brunner's glands, which secrete more alkaline mucus to help protect everything. And they are folded with the mucosa to make up the plicae circularis. So plicae circularis, see these structures, these are designed to increase surface area. You increase surface area, you increase the ability to absorb more stuff, which is exactly what we need. So we have each of these upjuttings, each of these folds, those are our plicae circularis, and that increases the surface area and absorption by 3 times. On top of those plicae, we then have villi, and those villi increase the surface area even more, as well as absorption by 10 times. And then finally, the microvilli located on the villi increase absorption by another 20 times. So it's all a numbers game. Oop. To give us as much surface area as possible in order to have as much absorption as possible. Here's what those plicae, these are the plicae, these are what these look like. So on a test, point to these. These are the plicae, and it's technically parts like this. All of this is more the mucosa layer, and the plicae itself, it kind of goes in between mucosa and submucosa. Then all of these things jutting out are the villi, and if we were to zoom in even further on these, we would have the microvilli. Here's the duodenum, a picture of all of this. We have those Brunner's glands, the mucosa, the muscularis, and then the pancreas close by. Here's another picture. We have mucosa and submucosa in the jejunum. Same structure, those plicae circularis, the villi, and the microvilli. Issues with the small intestine, we can get what's called celiac disease or celiac sprue. This is an autoimmune condition. Chances are you know somebody who has some type of form of celiac disease. This is specifically talking about gluten intolerance. So it's an autoimmune disorder where essentially the body is not able to properly absorb or deal with gluten, and it actually sees it as a threat and attacks all of it in there. So if you eat gluten, it sees it as a threat, creates an autoimmune response, and really just tears apart your intestinal tract. So you get lots of inflammation, and you're unable to absorb the proper nutrients. That is celiac disease. Okay, I'm going to go ahead and end part 2 right here, and we will finish out the large intestine and the rest of it in part 3. Thank you so much for watching, and good luck studying.